And recording has started. Okay. So today we'll continue on looking at the structure of DNA. And after the components were identified, right? So we know that we had nucleotides and we were able to see that you have these 4 bases. There was a lot of work that was done after that to figure out what exactly is the structure, how are these bases organized within the genome. And the work done by Chargaff looking at the composition of the different bases really kind of laid the foundation for Watson and Crick, right? Who were the ones who, the 2 scientists that actually, with the help of other scientists, Rosalind Franklin, that were able to come up with the structure of DNA, okay? And besides those composition from Chargaff's, right, where he showed that, the content of purines was approximately equal to the composition of pyrimidines, we knew that from Rosalind Franklin work, who was this physicist working at the time, she did some experiments looking to see whether or not she can figure out what is this structure of DNA that they have now. And based on her X-ray diffraction experiments, it suggested that DNA might be organized in kind of a helical shape. Okay, so it's a regular repeating structure. And with that information, then Watson and Crick were then able to say, okay, now we have the base composition, right? So we know what the nucleotides are. We have this information from Rosalind Franklin. How could we organize the structure? And you can see over here in this photograph, Watson and Crick looking at a kind of a model of what DNA is supposed to look like, or what they hypothesize that it might look like and how it's organized. So there's a YouTube video talking about the discovery of the DNA double helix. If you're interested, you could definitely check out this link. But together with Chargaff, Rosalind Franklin, and Watson and Crick, they really laid the foundation to show that the structure of DNA is in this helical structure, okay? And this DNA helix is kind of like in a right-handed structure. If you can look to see that, okay, it's kind of twisting in this order. And the way that the organization happens, you have 2 strands of DNA. They knew that you had 2 strands of DNA, right? And you had the curving side of the ladder, which represent the sugar phosphate backbone. So these are all the sugar phosphate backbones that you're seeing, right? Remember, you had the sugar group and your bases, your nitrogenous bases, which are your nucleotides. And the ladder rounds are the base pairs, right? So what is base pairing? A base pairing with a T, for instance, right? So you had these base pairing, and those were what's representing here in these ladder shape. And in general, based on the conformation of this helical structure, they were able to show that you had about 10 bases per turn, right? So here is, you can imagine that you're going through this turn, and if you count the number of bases, it should be roughly 10 per turn, okay? And also the critical information here was that the purine in one strand is always across from a pyrimidine, okay? And this is something that was discovered. I mean, you guys, again, all familiar with this. And this was important because the purine across from the pyrimidine was important so that the structure is not kind of bending or kinking or bulging particularly, right? So if you imagine if you had, 2 purines opposite each other, you would have kind of something that's sticking out here, and then it's smaller here if you had 2 pyrimidines next to each other. So this kind of setup was important for DNA structure to maintain the kind of conformation that it have in this helical kind of shape. So in addition, they also showed that, again, base pairing is important, as I just mentioned. So the base always pairs in a specific way. As I said, an adenine pairs with a thymine, right? So A always pairs with T, and a guanine would pair with a cytosine. On the outside, you have your sugar phosphate backbone and what you would call and then these hydrogen bonds that's base pairing the bases to each other. Okay, and this is something that was again important for stabilizing the structure. ANT always bonds up being this. It always creates a double bond, right? 2 hydrogen bonds. And G and C have 3 hydrogen bonds. And this is important because as you'll see later, you know, if you have 3 bonds versus 2 bonds, then it's a little bit more difficult to break those bonds than if it was just 2 bonds, right? It's kind of like, you know, if you have if you have multiple attachments versus just 2 attachments, right? I mean, you will be able to disassemble that, whatever structure you're working with, much faster. And the way that they visualize using some structural analysis, the way that the DNA could possibly be organized within the genome could take the form of any of these 3, right? And they're usually just referred to as A, B, or Z, okay? And the one that were proposed by Watson and Crick is really the B conformation, okay? Where you have this hybrid with a double strand nicely organized around each other. It is the conformation that's most often seen within the cells, right? But it's not the only conformation that might exist, right? So there's this other conformation that was discovered at the same time. And you would find that in most cases, when you have a stretch of DNA that's highly GC rich, it might form this conformation, which is in the Z form, okay? Quite often, this A form is usually found when you have a DNA RNA hybrid. So one strand is DNA and the other strand is RNA, and they're base pairing together, and that's the conformation. But in general, this B form is what's most commonly found with DNA hybrid formation. In addition to that, based on the way that these 2 strands of DNA is organized within the cell, this also provides information about how DNA might be copied, right? How might DNA be replicated, okay? So because one of the strands is the complement of the other, when you separate these 2 strands, right? So this is the complement of this, or vice versa, this is the complement of this, right? You see the base pairs, the A and T matching together. When you unwind this DNA, each of these strands serves as a template for a new round of DNA that's being made. Okay, so they serve as the template. You have these new strands being made, and what you have at the end of this DNA replication is 2 double-stranded DNA. One is from the original, and the other one is new. Okay, and we'll talk about DNA replication a little bit later, but this is really highlighting the semi-conservative nature of DNA, right? Because one, you're still saving one of the original strands. It's not completely conservative, right? So it's just semi-conservative. And this is something that they were able to discover just by looking at the structure, seeing how the base pairs come together, and how once you unwind that double helix in that DNA, how it enables DNA replication to occur. Okay. So because DNA is usually found in a double-stranded conformation, those strands, for DNA replication to happen, if you need to make a new copy of a piece of DNA, you need to be able to separate those 2 strands of DNA, okay? So to separate those 2 strands of DNA, we usually refer to this as DNA denaturation or DNA melting, or you're denaturing the DNA. And in order to do that, there's many ways you can do that within the lab or within a test tube. And based on different experiments from different groups, we know that there's many ways that you can do this. So one of the most common ways to denature DNA or to unwind the DNA and separate the strands, so you're breaking the hydrogen bonds between the base pairs, is by heating it, right? So high heat will break those hydrogen bonds that hold them together, and then, but not the phosphodiester bonds that's keeping the structure together, okay? So you can use high heat. Low salt concentration is also, can also denature DNA. And one of the ways that the low salt concentration is able to do that is that when you apply or when you provide within a test tube a low concentration of salt, right, it has a very low ionic strength. So because it has a low ionic strength, it is very negatively charged, okay? And DNA, as you know, is also negatively charged, right? So it's strong enough to kind of repel the forces between the two strands of DNA and break those bonds, okay? You can also use an alkaline solution such as sodium hydroxide, for instance, which is also used to help with breaking the bonds or denaturing DNA. And this is really done by the fact that these alkaline conditions, the molecules from these alkaline conditions are able to cover the basis of the DNA when you increase this pH, right? So high pH, you have lots of OH group, right? So let's say you're using sodium hydroxide, which is NaOH, right? Sodium hydroxide. And really, it helps to contribute the protons that you would need, and particularly for like guanine and thymine, where you will need to have a higher concentration there. What effect do these solutions have on RNA? A lot of alkaline solutions can denature or can denature RNA, and that's why it's not most commonly used for RNA, and that's why RNA is also more unstable, right? Because these alkaline solutions, in addition to breaking the hydrogen bonds, it really breaks the phosphodiester bonds as well, and that can also become problematic for RNA, okay? Because then you can kind of denature it pretty much. Another thing that you want to think about is, you can imagine, we haven't talked about how you might visualize DNA, but you can, if you have DNA that's double-stranded together, oh, sorry, I didn't, okay, so I see it. Okay, so Christian?
Yes, I was curious. Once you go through this process with the denaturation of the DNA, there's no way to actually have the strands come back together, right?
Yes, there is. So, and that's what we'll talk about next. So DNA can come back together, can renature, and that can be done if you slowly cool the solution down. Yeah. So you can have you can have them coming back together.
Okay.
Yeah. Okay, Shayla?
Could you just re-explain the alkyl conditions and how it unwinds it? Because I'm not understanding, like.
Okay, so with alkaline,
affects the phosphodiester bonds. Like, what does it not affect the phosphodiester bonds in the DNA?
Okay, so yeah, so sodium hydroxide, so alkaline solutions, let's say for instance, sodium hydroxide is usually commonly used. And the way that you use this is because it increases the pH, okay? And when you increase the pH, you're going to also remove the hydrogen bonds in DNA. But DNA is double-stranded, right? So you're removing the hydrogen bonds and you're separating the two bonds, right? RNA is single-stranded. So because RNA is single-stranded, you're not, instead of breaking the double bonds, it will then attack the phosphodiester bonds instead. Right? Because it doesn't have a double-stranded, I shouldn't say there's no double-stranded RNA that exists, but in general, RNA is a single-stranded molecule. So instead of, if you apply that, it doesn't have the hydrogen bonds like DNA to really attack and break. And so instead with RNA, it attacks the phosphodiester backbone instead.
Okay, that makes sense.
Okay. Yeah, sorry that wasn't clear. Yeah. Okay. So for the way to denature DNA, to figure out, how high of a temperature you need in order to denature DNA, there were several experiments that was done. A lot of these experiments, as you can see, was done in bacteria because it was kind of the organism of choice at the time to do a lot of these studies. And you can measure the denaturation of DNA based on using an absorbance, a machine that will absorb light, okay? And this works because nucleic acids in general is able to absorb light at a certain wavelength, okay? And that's really because of the electronic structure of their bases and how it is, okay? So you can imagine when again, if the bases are exposed, they can absorb light and emit light. And you can detect this using a spectrophotometer. When you have these two strands come together, right, you kind of quench that absorbance and you quench that amount of light, right? So the visibility is much lower, right? You're not gonna have a lot of light. As you denature those two strands of DNA and they become separated, then you have more light that's being absorbed, okay? And you can see this by looking at this graph. So what you have on the y-axis is the relative absorbance at 260 nanometers, and this is kind of the standard absorbance for nucleic acid and for DNA, and versus temperature, which you have on the x-axis, right? So you're looking at this black line on this graph, and what you see as you're increasing the temperature, the light is not changing, right? The absorbance amount is not changing, right? Because 65, 70, 75 degrees, the DNA is still in a double-stranded form, okay? Most of it is still in double-stranded form. And as this goes on, right, and then you get to, as the temperature gets higher and higher, it starts to unwind and denature, so you're able to have more and more light being absorbed, right? And that's where you see, if you're looking here, this starts to increase. When you reach the point where half of your DNA has been denatured, that is what we usually refer to as the melting temperature of that strand of DNA or that pool of DNA, okay? So the melting temperature is when about 50% of your double-stranded DNA has been denatured. And again, as you will continue to increase the temperature, you will see really, really high absorbance, and it starts to plateau because that's kind of at the point where almost all of the double-stranded DNA should be denatured by the time the temperature reaches 95 degrees Celsius, okay? And so you can denature DNA at this temperature. Does it all happen at once? It doesn't, right? So you can imagine in your genome, you have double-stranded DNA present, large amounts depending on the organism. As you start to denature DNA, and in this case, you will need to do this, for instance, maybe for DNA replication, right? This is when it naturally happens. If you put double-stranded DNA in a test tube, you apply these conditions, you're denaturing your DNA as well. It doesn't all happen at once, right? So it starts, certain parts will start separating faster than other parts. And more than likely, the parts that have more ATs available will start to break faster and starts to open much faster, right? So these are kind of the AT-rich regions. And another important factor to take into account, how fast you get to that 50% or half of the pool of DNA being denatured really depends on the GC content. As I mentioned before, G and C, the pairings contain 3 hydrogen bonds compared to A and T, which is 2 hydrogen bonds, right? So it just takes a little bit longer to break 3 bonds than it is to break 2 bonds. Yeah. And so the higher in general, the higher the GC content, the longer it takes, and also the higher temperature you need to get to this, to separate to denature the DNA. This is a graph, again, highlighting exactly what I did. And this is actually an experiment that was done looking at different pools of DNA from different sources, right? So from different bacterial sources, yeast, and also bacteriophage. And what you can see is that on the y-axis, you're looking at GC content, and on the x-axis, you're looking at the melting temperature. This was also done for, many of these other species which have relatively different GC contents as well, okay? And if you were to look at this red line only, that's going up here, you can see something that's AT rich very quickly. To get to 50%, it has AT rich happens very quickly at this point, okay? As you start going up in the amount of GC content, so let's look at the bacteriophage, T4. Well, this one is not listed here, but let's say we were looking at maybe Pseudomonas or Streptococcus pneumoniae, which is like 42% you have here. You can see that GC content is 42%. It lands here. The temperature is like about, 84, compared to yeast, which is Saccharomyces cerevisiae, 39, which is a little bit less GC content, so it falls a little bit lower, right? So it takes less temperature or a lower temperature to reach half of the pool of DNA to be denatured, okay? When you go all the way down to like Mycobacterium filii, which is like really high, rich in GC content, you can just see it just takes a longer time to get there, okay? So it really is a consequence of the GC content that will really determine your melting temperature of your DNA. So as was asked earlier, right? If you can denature DNA, can you also renature it? Yes, sorry, Shayla. Mm-hmm.
On the graph where it says ATDNA and it's like around like 65, is that consistent for every single like organism?
No, it's not going to be consistent for every single organism, because even though it says ATDNA, it really depends on how much ATDNA you have, really. So it might slightly vary, but it's usually within this range, right? It might slightly vary, but it's usually within this range, because maybe some organisms might have a little bit more AT, like you see here for the difference just between Streptococcus and yeast, right? They're slightly different. They're not that much different. It's a matter of 3 GC rich, 3 G plus Cs, the difference between these two, and you can see that there's a little bit of difference. So it just depends on the organism, but in general, highly AT rich is usually between 60 and 70 that you will see.
Oh, okay.
Yeah. Yeah, that's a good question. Thanks. So yeah, so as DNA, you separate DNA, you want them to be able to come back together depending on the situation. And depending on similar to what you will have for the denaturation, when they come back together, depending on how or what conditions you use to bring these 2 strands back together, most often you will have what you started with, right? Where you have the interstrand coming back together, so it's an intermolecular interaction. Or sometimes a single strand can bulge out, right? If the sequence is similar in a single strand and form what we call these intermolecular structures. And it just really depends on the DNA, the environment at that time, right? But the majority of the DNA that will be renatured will take this conformation. and this is kind of just what I said, depending on the condition, they will come back together. And that's really highly dependent on temperature, right? So the temperature, if the temperature is low enough that it doesn't promote denaturation anymore, but it's high enough to kind of weaken like transient bonding between like mismatched sequences, that's when you can have these short interstrand pieces of DNA, right? So it's low enough, there's nothing else to separate, but it's still kind of high enough where some of the two strands will still kind of stay a little bit separated if it's already been separated, and it will start cooling down and these two can come back together. Again, that's usually not most times what we have happening. Another way to prevent the DNA from coming back together is by rapidly cooling it, okay? So if you go from 95 degrees Celsius to 4 degrees Celsius very quickly, that also prevents renaturation from happening, okay? And some people might decide that, okay, you just take your DNA, you separate the strands, you stick it in a bucket of ice, you'll keep those two strands separated. And for other cases where you would like the DNA to be able to come back together, this is really based on how much DNA you have, right? And the higher DNA concentrations you have, the more likely those two strands will find each other and come back together as you start cooling the reaction down, okay? The other way that the renaturation happens is also in the context of time, right? So the longer you have, two separated strands of DNA mixed together under the appropriate condition, they will start coming back together. And if you have a higher concentration, it takes less time. You have a lower concentration, it takes a little bit of a longer time. And this is kind of highlighted here on this very similar graph as we saw where we were looking at the absorbance when the DNA was being denatured. You can measure renaturation in the same way, right? So you have the relative absorbance and you can do the time at 60 degrees Celsius, which is kind of the temperature where you want to start cooling the reaction down to get to about 20, 25 degrees or room temperature per se. So in this case, you have two traces, right? So the black trace is showing you what happens in terms of the DNA coming back together. So compared to the other graph we saw, relative absorbance is very high, right? Because these strands are separated, you have a lot of light, so it's very high. It starts decreasing as the strands are coming back together as you start quenching that light over time, okay? So you're looking at if you have a higher, the same DNA, have the same melting temperature, right? So they will be denatured around the same temperature. You have more DNA, right? 25 micrograms per mil versus 10 micrograms per mil. It's much faster, right? That they start quenching and coming back together versus if you have 10. So you have more DNA, it will come back together much faster under the optimal condition. So it's really concentration dependent, along with some other tweaks that you would do to the condition to make this come back together very quickly. So the question is, as you're thinking about this as you're studying, whether it's in the discussion section or you're preparing for the material for the exam or throughout the lecture, you want to think about things that might affect DNA, double-stranded DNA, okay? And we talked about how heat affects the DNA and what it causes, right? And it might seem very simple to you and very intuitive, but having these very basic understanding of what is happening will make it easier for you to answer a more complex question about what's happening at the DNA level. So I will leave this here. It's in the PowerPoint presentation as well, but just thinking, we just talked about it. So you guys can, if you want to just jump ahead and answer, and it should be very obvious to everyone, right? How does heat affect DNA duplex, right? So anyone want to guess?
It denatures it.
Right. So it denatures it. And you can see once it denatures it, right? This is going to become important as we start thinking about a little bit later in the lecture when we talk about PCR, right? And that's why it's good to just have this idea in your head already. And so the next thing is, you talked about DNA and how we refer to DNA, right? I mean, if you're not a scientist, you're, having to think about what is DNA, what are you referring to when you mention such a molecule? You can express DNA in many different ways, and it's expressed in many different ways depending on the field that you're in and what you're studying, right? But by far the most common way that DNA is expressed is by talking about it in terms of base pairs, right? So how many nucleotides do you have together, okay? And because a nucleotide is double-stranded DNA, they're pairing with another base. So they're referred to as the number of base pairs that you have. And you can also refer to DNA in terms of the length. 10.4 base pairs, which is one of the helical turns we just talked about, is about 32.2 angstrom, right? In terms of molecular weight, where if you're working in biochemistry or you're a biochemist, you tend to think a lot about DNA in terms of molecular weight, right? So 660 is the molecular weight of one base pair. Yeah. So it just depends on the context. I think for the purpose of this class, a lot of what we will be thinking about in terms of nucleic acids and how they might be expressed will be in terms of base pairs. Mm-hmm. You can measure the amount of DNA and the length of DNA using many different methods, right? So it can be measured by an electron microscope where you can visually look and see what you have and quantify that. Or you can run a gel, and we'll talk about this method because this is the method that's most commonly used in molecular biology, gel electrophoresis. So now that we know, okay, we have DNA, what's the structure of DNA, how the DNA, double-stranded DNA is organized within the cell, we think about it more on a global scale, like how much DNA do you now have within the genome? So you would predict that an organism that is very complex, right, will probably have more DNA, and such, because it have more DNA, it have more genes, right, than an organism that's a little bit more simple, okay? So the genome size of an organism is defined as the total amount of DNA contained within a haploid cell. And that is usually referred to as the C value, right? When you think about genome size, the C value, the amount of DNA. So as I mentioned, you have larger organisms such as mouse and humans, and they will have more DNA, right? They're just larger organisms. They'll have more DNA, right, than you would find in yeast, which is smaller, it's a smaller eukaryote. And they do, in fact, have more DNA than the yeast cells. Yeah. When you think of yeast, right, compared to bacteria, so yeast is a eukaryote, bacteria is a prokaryote, it's a little bit more complicated. It has more DNA than you would find in bacterial cells. Okay. However, the DNA amount within a cell doesn't correlate with the amount of genes and the complexity of the organism per se, okay? So if an organism is with much more complex, for example, if you're thinking about maybe, let's say the frog, for instance, right? So the frog, it's a frog, right? I mean, we would tend to think that humans are way more superior than a frog is, right? or a little bit more complex than a frog is, right? But the frog has 7 times more DNA than you would find in a human cell, right? This is same for a plant. If you think of lily, for example, which is just a plant, we all see lilies growing everywhere. It has 100 times more DNA per cell than a human. Okay, so there's these paradoxes that we refer to where it's not always the case that you have a more if you have a more complex organism that you will have more DNA within the cell. Okay, another correlation to this is that, so what about the genes, right? Are the genes also correlating with the organism complexity? And we know that that's not the case, right? Because again, we have genome sequences from many, many organisms now. We know the amount of DNA that's present within those genomes. We know the complexity of those genomes, right? And we also see that the gene content, the amount of genes that are present within those organisms, do not correlate with the complexity of the organism, right? And that is kind of from a lot of comparative analysis studies, and it's holding through for the most case, even now as we're sequencing, individual genomes within the same species, for example, right? It's very similar. So in addition to that paradox where, you have a greater complexity, you might have different amount of DNA within the cell. Here we just have examples of 4 different organisms, right? The worm, which just seems, it's just a worm. It is complex in its own way for different purposes, but of course it's not as complex as humans, right? You have fruit fly. A lot of genetics is a very useful model for studying for genetic studies. Rice, which is a crop, a very important crop for agricultural purposes. And then we also have humans, which we know, we are complex in ourselves, right? So there's lots of different degree of complexity depending on what aspect you're looking at. The genome size, so for these kind of correlates a little bit with the complexity, right? So the worm is a little bit smaller, fruit fly is a little bit larger than worm, and you can see rice is a little bit about 3 to 4 times larger, and then you go on until you get to humans, which again, larger genome size. And this is written in terms of millions of base pairs, right? And that's what this MBP means. We said that DNA, we refer to it in base pairs, and you will see an M in front of it if they're talking about millions of base pairs. So here you have 100 million base pairs present within the worm genome. So when do we think about, what would you assume based on this in terms of gene content, right? What does that mean in terms of gene content for you? Like just as a guess,
how many genes are there?
Pardon?
Like how many genes are there?
Yeah, like how many genes would you expect to find, in these different organisms? Let's say with worm, how many genes do you think that's present within the C. elegans genome? Just ballpark. I can tell you it's thousands, so that's a weird place.
It will be the lowest, but I'm not sure about the exact number.
Pardon?
Like my guess.
It will be the lowest.
The
number?
lowest? Okay.
Yeah, for the worms, right?
It would be lowest, okay. And in terms of, someone was saying the numbers?
13,000.
13,000? 13,000, okay. What about fruit fly? So let's say that's 13,000. What about fruit fly?
27,000.
Okay, 27,000, okay. And rice?
200.
Random guess. If it's larger, right? So far we have 13,000 for C. elegans, we have 27,000 for Drosophila. So rice would be, you think it would be a little bit more?
50,000.
A lot.
Pardon?
50 or 200,000.
200,000? Okay, and that's fine. And what do we think about humans? How many genes do you think we have in the human genome that you guys, how many genes do you have?
Only 20,000.
Around 20,000? Okay.
Yeah, that's it.
Okay, so great. So I mean, these are all good guesses, right? I can tell you right now that between C. elegans, rice, and human, the amount of genes is very similar. Okay, so let me see if the numbers are going to pop up here or not. Okay, I think I have it as a quiz question for you guys. So that's why the numbers are not there. So you can just take it down, and you will also hear it in the recording. But C. elegans have about 20,000 genes. The fruit fly have about 14,000 genes. So the genome is larger, but it has less genes than you find in C. elegans, in worms. Rice have about 30,000 genes, so slightly larger than both of these organisms. But then the human genome, which is humans versus plants, right? It has about 25,000 genes present within the genome, okay? So you see this huge variation of genome sizes, and this is true if you look at many other organisms, the amount of genes is very similar to across genomes, right? So what is if they have a larger genome size, they have more DNA, they're more complex, but then the gene content, which is kind of the major hub for regulating many things in our body or in the organism, why is it that you have similar number of genes across these very different organisms? And that really relates to, and we will get into that as we talk about transcription, right? You will see that even though a lot of these genomes have more DNA, a large portion of that DNA does not provide information that encode genes, right? So that's why you have less genes. There's a lot of other aspects of a genome that are important for the survival of that organism, but it doesn't encode genes. There's also the process during when we talk about transcription, where even if you have the similar number of genes, you can make a variety of different proteins, right? And this is really based on how the mRNA gets processed, okay? So we'll talk about, going from DNA to RNA when we talk about transcription and what happens at that transcription step that can promote more proteins for the same set of genes, okay? So as I mentioned, you have the way that a lot of this information is stored will determine what gets in the end, what gets produced into a protein, okay? So how many genes are encoded in all of the DNA within a cell, right? And how much space is within the genome and the DNA, right? So what is the spacing between genes, right? That also will affect how some of this information might be stored and also used. So in order to produce a protein, you really need to go through two important steps, right? So you have to go through transcription, right? So you have your double-stranded DNA. I mentioned RNA is a single-stranded molecule. So you're transcribing the information for this gene into a single mRNA molecule. And then the additional proteins that are involved in this process will then read this message and then translate it into a protein depending on what this message said, okay? Again, this message is just coming from what's encoded in the DNA. And that is really important in terms of understanding how information gets propagated within the cell. Encoded in this DNA is everything you need to make a protein. 3 nucleotides determine the amino acid that's going to be used, right? So A, T, and G, there's only 3 bases that will determine that you will make a methionine, okay? And also you have, as we will talk about transcription, the strand of DNA that's used to be transcribed is important, right? So the strand that's always transcribed is the non-template strand, right? Because it's very similar to the information that's being encoded by this strand, okay? So it needs to provide this information. This is also important, as you will see in transcription, because of the way that the enzymes that are performing the transcription work. Okay, and so I'll just keep this information as we proceed, so you will get to that once we discuss it. And as I mentioned, the codon, every amino acid that's encoded within the proteins is determined by 3 sequences or 3 nucleotides. Okay, and depending on the amino acid, there's only 20 amino acids that's present. There's no more, there's just 20, okay? So those bases, those nucleotides have to provide information for each of those 20 amino acids that are important for putting the protein together. We'll talk more about this as we talk about translation, but this is just to kind of get you familiar with this. And when you think about the way that the genes or the DNA is organized, I guess, into a gene structure, there's many different parts that we will be discussing in the upcoming lectures, okay? So this is double-stranded DNA. We will talk about the fact that there's always orientation. Actually, we already spoke about the fact that there's always orientation, right? We talked about the fact that you read your DNA, you have that 5' phosphate that's always available and 3' OH that's always available, right? So there's always an orientation to DNA. This is true for RNA, and this is also true for proteins, as you'll see, okay? So double-stranded DNA, 5' to 3', the strand that's complementary will be 5' to 3' to the bottom strand, right? So that's kind of the matching. Gene structures have transcription start sites. Where are you going to start transcribing to make this RNA? What information is being encoded, right? So this is an open reading frame, right? So open reading frames are usually called open reading frame because it doesn't contain any stop codons to interrupt the translation of this region, okay? And it is called a reading frame because ribosomes that are involved in translation can read this information in 3 different ways, okay? So that's why we refer to it as a reading frame. You usually have a start, you have a stop. This is the open reading frame. And then at the end, you also have a series of nucleotides towards the end that are involved in terminating transcription, okay? Yes, questions?
So the open reading frame, that's the only, so the transcription initiation site, that's what tells the ribosome to start here. And then the ORF is what actually is read to create what is needed to make the amino acids.
Right, so this open reading frame is what will tell the ribosomes you start translating here. So it'll start, say, okay, I start translating at this ATG, that is the first methionine, right? GGA is the next amino acid. And it will know that, okay, this is the only information that's encoded in that gene. So this is the protein I need to make.
Okay.
Yeah.
Thank you.
Mm-hmm. So that's kind of, and then this is, again, this is what gets translated. Even though all of these additional regions get transcribed, they're not translated into a protein, but they're important for the mRNA to be processed and become stable, et cetera, before you can even initiate this translation state, right? Because if you guys recall, transcription happens within the nucleus and then needs to be exported into the cytoplasm for translation to happen, okay? So having these additional sequence you will see is absolutely important for the integrity of this RNA later on. Okay, there's a lot of regulatory information here, but they don't get converted into the actual protein and translated. Do you have another question, Christian, or your hands are still?
Oh, no, sorry, I forgot
Yeah.
to.
Just thanks, yeah. Yeah, so then that's, we talk about protein structure. I'll try to speed through this because we'll get back to this later on, okay? So proteins, again, small, not small, they're chain-like polymers of small subunits called amino acids, right? For nucleotides, you have 20 different amino acids. There's also a structure to the amino acids, which we will get into in more detail. And pretty much, you can think of the general structure of amino acid, right? It has an amino acid. Usually it has kind of a hydroxyl group. Hydrogen is usually at one of the sides. And depending on what is added, this amino acid gets added to this structure will determine the amino acid that's there, right? So it's the amino acid side chain that determines what the amino acid is and just the structure of these, right? But you can see that this base is really this amino group is very consistent for all the different amino acid, and it's just what is added to the side chain that really makes the different protein or give them their unique character that they have. We will learn about the joining of amino acids, right? The bonds that are formed to create these a string of to make a protein, so create a string of amino acids, which is usually referred to as polypeptides, right? And a protein can have one or more different polypeptides, similar to DNA that have directionality. Proteins also have directionality, right? So you usually have what we refer to as an N-terminus, right, which is the amino terminus, or we also refer to the other end as a C-terminus, right, where you have this kind of carboxyl group at the end here, okay? So there is a directionality to amino acids in general. And you'll see why that's important again a little bit later when we talk about translation. So that's kind of all I want to talk about DNA before we get into cloning. Any questions about the material? Okay.
Yeah. So earlier when we were talking about the DNA cooling down and how they like can attach upon itself with the, what was it, a bulging or whatever you called it? Is that it like folding upon itself, like that single strand?
Yeah. So the single strand will fold upon itself. So it's really, imagine that you have a stretch of DNA and then a few base pairs downstream, you have sequence that is complementary to parts of that single strand DNA. They can fold over and make a hybrid. Yeah. This is actually, this happens with DNA, but you'll see it really happens a lot with RNA because RNA is just always in that single stranded format. So forming these, what we call secondary structures and intramolecular secondary structure happens, yeah. And it's just based on similarity and again, based on the conditions that this allows this to happen.
I have another question. Yeah.
Yes.
When you were talking about denaturization earlier, could you re-explain how low salt concentrations cause the DNA strands to split apart?
Okay. Oh, okay, here, where we mentioned the low salt concentration.
Yeah.
So you remember I said DNA is negatively charged. And when you provide a low salt concentration to the solution, what it does is that it removes the ions that are shielding those negative charges on the two strands of the DNA from each other, right? Because imagine, because DNA is negatively charged, if you have two negatively charged molecules, they're going to try to repel each other, right? But usually those negatively charged get shielded, and that's why DNA is able to come together, right, and form a double strand. However, if you lowered, and again, this is all because the conditions even within the cell or within the test tube is optimal to allow that to happen. However, we can force that separation by lowering the salt concentration because salt will remove those the shield from those negative charge and then Now both strands become, their negative charge get exposed, and then they start to repel each other.
Okay, got it. Thank you.
Mhm.
I have a question too.
Yes. Mhm.
So we've seen that some species have more genes than a human, although they are way less complex. So what's the reason again? What are these extra genes for?
Sorry, let me turn, I'm sorry, could you just repeat that one more time? I'm sorry, I didn't hear you.
Yeah, sure, sure. I
Mhm.
mean, I was saying like we've seen like some organisms have more genes than a human. Yeah, like in this
Uh-huh.
slide, let's say
Mhm.
like the I think rice,
Mhm.
also. Yeah,
Mhm.
although they are like less complex. So what's the reason again?
The reason is just it's really over evolutionary time, right? So these genes just got conserved, the amount of genes got conserved over evolutionary time and is maintained. But the organism is a little bit more complex, and the complexity is really driven by the other regions within the genome, and also by the fact that even if you have the same amount of genes, the versions of proteins that you can make for that same, for a more complex organism is much higher. Okay, you can make much more complex. But in terms
Is it
of the
because of
number of
splicing?
genes, if you go away, it's just the nature of evolution, right? I mean, at some point, you know, I don't want to get into for this to be an evolutionary lecture, but at some point, you know, the organisms will have probably similar gene content, and that similar gene content just get made, right? You're not acquiring new genes unless something happens to your genome. Maybe your genome got duplicated or something to increase the amount of genes, right? But the DNA can be increased because of what we will discuss a little bit later, because of, like, for instance, things like transposable elements, right? If you have transposable elements are pieces of DNA that can make many, many more copies of that piece of DNA, right? So you can expand the genome significantly, but the amount of genes will remain the same.
What about splicing? Does that contribute
That's
also to-
exactly the reason. That's exactly what that's exactly right, yeah? So even though you have the same number of genes, you're more complex, and you can deal with that complexity and deal with the many different processes because of splicing, right? So
Okay.
what splicing does, it is that it creates these alternatively spliced versions, and each alternatively spliced version will encode a different type of protein, right?
So complexity is determined more by the number of proteins rather than the number of genes. We could put it this way, right?
Yes, can become by the number of proteins, but it's just not proteins alone. As you'll see, there's other elements in the genome, non-coding elements that doesn't code for gene, that also provide that complexity.
Mm-hmm.
Yeah.
Okay, yeah.
So, and in terms of, you know, with the proteins, right? You can imagine within the cell, you have your genes. I mean, alternatively spliced events is kind of controlled. So it doesn't randomly happen, right? So depending on the organism, you might have more alternative splicing, maybe like during a certain developmental stage. Or, for example, in plants, you have more alternative splice happening, or alternatively spliced events happening in response to certain environmental stresses. So it really depends on the environment and also the development of the organism and certain other types of processes. So sometimes I would say that it's a little bit difficult to predict how many proteins is going to be produced at all times, because you just don't know, right? So that's why I'm a little bit hesitant to say it's really the complexity of the proteins rather than you know, other aspects of gene regulation that might be happening.
Yeah, that makes sense.
Great questions, guys. Thank you. Okay, so then we're going to talk about cloning. We don't have a whole lot of time, but I'll try to see how far we get. And because this is kind of I guess it's kind of the core of molecular biology, as you will see, is understanding cloning, right? Because that's how we are able to even understand how a gene functions, right? We have to be able to clone it first. Yeah. So we will talk about restriction enzymes and other important enzymes such as DNA ligase, vectors, and then also the technology that allowed us to even be able to do any kind of cloning or the advancement of technology that allowed us to do even more sophisticated cloning, which is PCR and reverse PCR and stuff like that. And, so imagine, back in before cloning times, right? So let's say in the 60s, right? Or even in the early 70s, right? Say you want to study a gene, a human gene that's involved in some kind of growth process, for instance, right? So the human growth hormone, for instance, gene. You want to know what is the sequence of the gene. You want to know what the promoter of this gene look like. You want to know where does RNA polymerase bind to be able to transcribe it. You want to know what happens to that gene to cause dwarfism, right? Because this gene is involved in dwarfism, right? So all of these questions you need to answer, right? How would you go about answering that? Imagine someone say, okay, tell me what the function of, I know that there's a gene in your genome that does this particular function. How would you go about studying that?
Studying the gene itself?
Yeah.
You would need extra copies of the gene because you don't want to just accidentally ruin the one copy you have.
Right.
The process is PCR, I believe.
Mm-hmm. Exactly, right? So you need to get that gene, make more of it, right? And one way you make more of it is by PCR, right? So, but back then they didn't have those technologies. So it's kind of like what they have been able to achieve and for us to understand with not having those technologies available to them is actually quite amazing, right? So initially, what they wanted to do is that you have to get the gene out of the genome. So you have to get it out of the genome. So it means you have to clone that human growth hormone gene, okay? And cloning at that time requires, the classical term is that you're making a recombinant DNA molecule, right? And then you're putting it into the cell and maintaining it in that cell, okay? A recombinant DNA molecule usually means that you're referring to two different parts, right? And you're putting them together, right? So they're not from the same source. So you need a vector, which is a DNA molecule that contains the information needed to propagate that cloned DNA into the host cell. And you need the piece of DNA that you want to propagate and maintain in the cell, and that is usually that gene, okay? Once you put it into the cell, bacteria, so bacteria can only replicate and propagate circular plasmids or circular pieces of DNA, okay? So once you put that into the bacterial cell, the bacteria will recognize it. It has an origin of replication. The bacteria is gonna make tons and tons of copies, right? And you're gonna just make more and more, right, of your clone that you want, right? And that way you can study it easily, right? So in terms of, you can, in plants, you can literally make a clone by cutting or growing. You can cut a piece of the plant. You guys see that you can cut a piece of a plant and then just generate new plants from that, right? You can also make a copy of a plant or a clone version of a plant from a single cell, right? This is also possible. You can clone frogs, right? There's a lot of work in research where there's frogs have been cloned. And you guys can recall from in the 1990s where they cloned the sheep dolly, right? And you can even think if any of you are identical twin, an absolute identical twin, you're kind of considered a natural clone, right, of your sibling. Yeah. So cloning is important for us to be able to study gene function. The way that cloning was done with very limited technology at the time was using restriction endonucleases, okay? So restriction endonucleases were first discovered in bacteria, And there are enzymes present within the bacterial genome that wants to prevent invading or foreign DNA from propagating in their genome. So they just cut it out, right? So they're called restriction enzyme because they're restricting the propagation within the bacterial cell, right? So they're restricting the propagation. These enzymes cut within whatever foreign DNA. So that's why it's called endo, right, for within. If it's called exon, which you will see as we talk about other types of enzymes later on, they will cut or destroy outside of DNA or any kind of nucleic acids, right? So they're also very specific, right? So they can cut, they only cut at very specific sites. So they recognize certain nucleotides and this is where they cut. So they're kind of, we refer to them as molecular knives, right? Because they're really, they're very specific in that sense. Restriction and the name that we have from restriction endonucleases is usually come from many different sources of the microorganism, right? So usually they're named depending on parts of the name of the microorganism. So in this case, let's say we're talking about, this is usually referred to as Hindi3, right? So you'll hear people refer to this as Hindi3, that's the name, the common name that's being used. It came from Haemophilus influenzae, right? So H is the first name from the genus, which is Haemophilus. The next two letters are the first two letters of the species name, right? So in here, so HIN. And then sometimes there's a strain designation. So within this genus, there's multiple strains. And depending on the strain, you will add a letter for that as well. So that's HIN, okay? If the microorganism produces more than one restriction enzyme, so let's say Haemophilus influenzae produces many different restriction enzymes, then you will add the number to whichever one you're referring to, okay? So if there's a Hindi 1, there's a Hindi 2, and there's a Hindi 3 in general for this particular type of restriction enzyme, okay? So you can have multiple letters associated with it. And as I mentioned, they recognize very, very specific DNA sequences and cut only at that sequences, right? And this is important because they have to be precise because these are produced in bacteria, right? So, and they're used to restrict invasion of foreign DNA. So you don't want to make a mistake because you also have DNA within your genome, and we'll talk about how they actually prevent their own self from chewing up their own DNA with these endonucleases, right? So these restriction enzymes can recognize 4 base pairs, 6 base pairs, or 8 base pairs. And these sequences refer to they're palindromic, right? So they're read the same way from front to back, right? So AGCT is AGCT, okay? So depending on how so these you're just seeing the single strand here, right? But imagine that it's a double-stranded sequence. DNA is always double-stranded unless there's a reason for it to be single-stranded, right? So there's always 2 strands. And depending on the sequence, these enzymes will cleave between very specific bases, okay? So that is also very specific. So each of these are very commonly used restriction enzymes in the lab, and they will recognize certain sites, right? So they recognize this sequence. So it will recognize, let's say, GGATCC, but it knows it will only cleave between these 2 G's that's present here, okay? But the actual sequence that's recognized is this entire sequence, okay? So that is kind of one of the advantage of using restriction enzymes, that it's highly reproducible and highly specific. There's different types you can have other types of enzymes where you have 2 restriction enzymes that are referred to, they're kind of identical in their function because they recognize different sites in the same sequence, right? So SMA1 or SMA1 and XMA1, right? You see the sequence is the same. There's 3 Cs, 3 Gs, 3 Cs, 3 Zs, 3 Gs. However, they cleave between the 2 different bases, okay? And these are usually referred to as heteroschizomers, okay? So they're SMA1 will cleave here in between, and this XMA1 will cleave here, okay? There are some enzymes that will cut the same site in the same sequence, and those are usually referred to as schizomers. So you could have another enzyme that will recognize this site and actually cut right here as well. Right? So those are also referred to as isoschizomers. And the way that the restriction enzymes are able to recognize the site, right? So you have double-stranded DNA. Your restriction enzymes are going to be there. They're going to scan along the DNA, looking for the site to recognize. And this, again, imagine this is an invading DNA in this context, right? So the restriction enzymes are there. They're going to scan along, looking for the sequence within that foreign DNA. And it will bind and attach to that particular sequence. Once it attaches to the very specific sequence, the binding is much stronger. It starts to bend the DNA. Bending the DNA starts breaking the bonds in the DNA, right? And the enzyme will perform the nicking or cutting of the DNA, okay, and separate the two parts of the double-stranded DNA. Okay, this is kind of the way that it does. Again, trying to find it again, scanning along and identifying the specific site that it needs to cleave. Structurally, right, you can see this if you're looking under like some kind of X-ray crystallography image or some kind of like a high resolution X-ray crystallography image. You can see how it's grabbing the DNA, right? So the blue hybrids, helixes, right, that's your DNA, right? You have your major groove, your minor groove, and you can see that this is an enzyme where it's kind of grabbing both the minor groove and also interacting with the major groove, right? So it's really grabbing onto that DNA once it's bound, yeah, and helping to bend it. Yeah. How does it cut? So restriction enzymes can create 3 types of three different versions of that cut DNA. They can generate what we refer to as 5' overhangs, right? 3' overhangs, or they can cut a more blunt end. Okay, I'll explain the blunt end first because it's just easier. So you have your double-stranded DNA, right? XMA1 cuts here on both sides of both strands of the DNA at the same site. You separate the DNA, it's a clean break, right? There's no base that's remaining like hanging over. However, with a 5' overhang on cleavage, and in this case, let's say it's E. coli 1, it makes a staggered cut, right? So it cuts between G and A only, right? So it will cut the G and A here, and it will cut the G and A here. That's the cleavage site. Because it cuts in such a way, when you separate these 2 fragments, you have this overhang, which we call as a 5' overhang, right? Because again, orientation of DNA, always remember that. You have the 5' hanging over this, but this other part, you have the 5' hanging over here, okay? So it's kind of hanging over. The other enzyme, if you have, for instance, PST1, you cleave here, on these sides. Now you have the 3' that's overhanging, okay? So these types of cuts is also referred to as sticky ends, right? Because the ends can easily stick back together, right? So they refer to as sticky ends or the particular 5' overhang. The type of overhang you have is super important for your next step. Where is it that you want to use this enzyme to be able to clone something, right? And that's really important, the type of overhang, because you want to make sure whatever you're putting within, whatever you're going to be putting between these 2 parts of DNA needs to match what is overhanging here and what is overhanging here, okay? So that's super important. And we'll have some practice examples of that in the next class, right? So we can kind of make sure that you get that. So I think it's 1:50. Let me see. Yeah, we haven't gotten that far, but okay. So maybe let me stop here, ask questions. I'd rather go much slower through this process because it's a lot of information and very important information. So I will stop here. So you guys, if you have any questions, again, I'll stick around until 2, like 10 minutes, so you can ask questions if you have any.
I don't have a question, but I just kind of have a recommendation. I kind of found myself like zoning out here and there. I felt like you were talking a little slow. So maybe next time, if we could go through the material a little bit quicker. That's just how I feel. And I just want you to know that.
Okay, thank you. Any other comment?
Professor,
Yes.
I actually like your pace.
Yeah. Okay. Thank you. Thank you. Yeah.
Yeah, Professor, I think your pace was really good and your explaining was really good as well.
Okay. Thank you very much. Yeah. I appreciate the feedback. Yeah. Yeah. I mean, I think I think it also depends. I wonder, I mean, if you work in a lab, for instance, a lot of this information might be kind of, okay, yeah, I know this or redundant, but it's really very fundamental information that I think you need to get right now in order to deal with the rest of the material. So I feel like it's a lot of information that could be very confusing if certain steps are not done. So I don't know. I feel like it's some of the information, even though for some people it might be redundant from previous classes or from their experience, their work experience. I think it's kind of important to get it. Yeah. Hi, Sydney.
Just a quick question for like the 5' overhang. Will this be like an exam questions where you post like problems on how they're cut and where to match them?
Yes. Yeah.
Okay.
This will be like an exam question, and you probably will see. I think your TA will also go over some of these examples with you as well in your discussion sessions.
Oh, okay. Thank you.
I'm trying to see. Oh, Israel is here. Hi, Israel.
Hi, Don.
I had a question actually. I think, yeah, someone asked in the beginning whether or not, because I don't know if it was really clear in the syllabus, but your Zoom link for your TA is the same for your office hours is the same as the link for the discussion session, right?
Correct. Yeah, it's the same.
And do you have an individual iLearn setup or everything goes through my iLearn? That's what I wasn't sure about.
I have one for each section. So there's an iLearn page for every section. I also have one.
Yes, maybe you can post it there. I think just in case they don't see that information in the syllabus or don't get it.
Okay.
How are the discussion sessions this morning?
They were good. Just to clarify, we won't be discussing the new material today because I didn't have time to see what you guys got through. So we'll be discussing last week's material, and then the following week we'll get everything from this week and then the next week. So whatever you discuss today or before the discussion, we'll discuss that. But if you have questions, we can kind of answer them if we have time, just because it's only 50 minutes and we have a quiz to take. So
you. I think people are heading out a bit. Mhm.
Thank you, Professor.
Thank you. Mhm.